But so I take it we're in the middle of you guys' basic science, translational science, whatever module, is that correct? Okay. Yeah. So I remember when I was a fellow and I was sitting through these, and I think these are the most difficult ones to listen to and give us for the fellows, because after you guys aren't research oriented, it's not really the most clinical PowerPoint. So what I'm going to try to do is I've been given a task to talk about carcinogenesis and genomic profiling. And so, I'm going to be talking about a lot of techniques that we use or assays that we use. And I'm trying to show you how they're used clinically. The objective of this talk is not to make you guys experts at gene expression profiling or sequencing or anything like that, but just more to give you guys an overview so that you understand, how we use this in the assay and how they can affect you in the future. Now, the other thing we know what we're supposed to do is we're supposed to make 3 board questions, and I can tell you right now, the things I'm going to present today, none of this stuff is going to be on your board. But unfortunately, I have to give 3 board questions, so I'm going to make up some board questions that are relevant to your boards. And as you guys, I know most of you guys, I'm a GI oncologist. A lot of this talk and what I talk about is going to be GI oncology related. So let's go to board question number 1. 44-year-old male with newly diagnosed colorectal cancer undergoes a hemicolectomy. Test reveals T3 lesion, moderate differentiated, no evidence of lymphovascular invasion or peritoneal invasion, 0 out of 9 lymph node negative. Okay? This is a first-year question. First-year fellow. Who wants to answer this? What do we got? Chris? Let's go. What do you want to do?
I guess I would hope there were more lymph nodes, but if there were 15 and there were 0.
Okay. Anybody else? Okay. So, I think you bring up a key point, and a lot of this is going to be on your board. So the answer to this question will actually be number 2, adjuvant therapy, because as Chris brought up, this patient was inappropriately staged. And if you're inappropriately staged as a stage 2 colorectal cancer, the recommendation is you treat them as a stage 3 and give them adjuvant therapy. Okay? So the key point of this board question is you got to know your staging system, especially for the boards, okay? Especially testicular cancer and the cancers that we don't see. There will be many questions about what stage this patient is and how to treat them based on the stage. Testicular cancer, you guys better know your stage 1, stage 2, because how you treat them is going to be based on that. Melanoma, they will ask you questions like, if the depth of melanoma is 4 millimeter, what depth is that, what vessel stage is that, and what kind of margins do you use? So the important thing about your board is you got to know your staging, because they will help you answer your board questions. So let's talk about genomic profiling, what I'm going to talk about today. So what is genomic profiling? So genomic profiling is information about all the genes in a tumor cell, including variation, gene expression, etc., and the ways these genes interact with each other and with the tumor microenvironment. So as you guys know, kind of where we're headed toward is, we just don't want to treat patients with what they're diagnosed with, but we want to treat them based on what their tumor suggests us. So I like to show this slide because this is the old perception of what is cancer, right? So we all know we identify a patient with a mass, and the first thing we do is get a biopsy, right? Because we know no meat, no treat. We need some kind of diagnosis. So we get a biopsy. We take it to our pathologist. Our pathologist does a great job of looking at the microscope, and if it's breast cancer, if it's lung cancer, or colon cancer, you're going to treat it each way. The problem with this schema is currently we don't care what's the underlying biology. If they're colon cancer, it doesn't matter what kind of colon cancer, I'm going to treat them with colon cancer regimens. And the question is this the way we should be treating them? Now, as you know, we're entering the era of personalized medicine, and you guys have heard this word been thrown around multiple times for the past year. And what is personalized medicine? Well, Richard Siskey, ASCO president, chair COGB, made this comment that knowledge of the molecular profile of the tumor is necessary to guide selection of therapy for patients, right? So what does this mean? It means that currently, what do? If we have one patient group, which is all colorectal cancer, I'm going to give them the same treatment regimen. If they come to me and they have stage 4 colorectal cancer, I'm going to talk to them about whether or not they should get FOLFOX or FOLFIRI, right? I don't care what type of colon cancer it is. I just know it's stage 4. I'm going to treat them. What I do know is that some drug is toxic but beneficial. This is okay, but our patients are sick. Some drugs are toxic and not beneficial, and this is bad because the patient is going to get sick and they're not going to benefit. Drugs not toxic and not beneficial, not good. And kind of what we're trying to find is what is a drug that is not toxic and beneficial. And as you guys have heard, even all the first years, this is personalized medicine. So what is the current model? Well, how do we treat patients, right? Well, we take patients, and you guys are here, well, we need evidence-based medicine. We need a phase 3 trial. We need to know what the clinical trials show. So our current model is we have patients, we enroll them in a clinical trial, they get either drug A or B, and whichever does better becomes the new standard of care. So in this case, drug A, because there is a line between A and B in the Kaplan-Meier survival curve, becomes the new standard of care. What are we trying to get? Well, this is more of the personalized medicine model, right? We take our patient, we do a molecular analysis of the tumor, and based on the molecular analysis, we decide the choice of treatment upon the molecular profile. So this is nothing new to you guys. This is our personalized medicine model, and the question is, how do we get there? Well, the simple thing is tumor biology, right? If we know the tumor biology, we know everything we need to know about the patient's tumor. And you guys all know this. We all learned this in 9th grade biology. The central dogma of molecular biology and what defines everything we do is 3 simple things. DNA, which is then transcribed into RNA, which is then translated into protein. So to know the tumor biology, there's 3 simple things we have to learn. But you guys know the problem with this concept is this becomes this, right? Now, all of a sudden, the question then becomes, how do we start dissecting out the integrated signaling pathway that defines the tumor biology, and how do we begin to start understanding how these pathways work together and separate against each other, with each other, to identify the best treatment for our patient. So the good news is we're doing a lot better than we were 20 years ago. When I was in grad school 20 years ago, we didn't really have a lot of things to study DNA, RNA, and protein, but now we have all sorts of things, right? For DNA, we can do next-gen sequencing, we can do chip analysis, GWAS, comparative genomic hybridization. For protein, we have antibody-based proteomics, mass spec, RNA, we can do RT-PCR, microarray, RNA-seq. So as you can see, the advantage we have now is the technology has advanced in such a way that we have many assays that we can use to study tumor biology, and the goal for us, and for you guys especially, is that we have to figure out how to integrate all of these new technology, which is going to give us this vast information about the tumor biology into our clinical care. So, new perspective, we have colon cancer, and instead of treating somebody with colon cancer with FOLFOX, FOLFIRI, Avastin, which we like to do in first line, is how do we do the molecular profile, right? We want to get to a point where we take this tumor biology and we want to know KRAS mutation, BRAF mutation, microsatellite stability, so forth and so forth. And so where we end up is instead of colon cancer being treated like colon cancer, we started treating colon cancer like the molecular profile. Okay, so there's a lot of topics to talk about, but I'm going to focus on 3 things because I don't want to go through every single thing that's clinically applicable. So the 3 things I'm going to talk about today in terms of genomic profiling and hope to give you guys information and clinical applicability is 3 things: real-time PCR, RT-PCR, microarray, and sequencing. Okay, so let's talk about PCR, real-time PCR first. So this is a basic schema of PCR. Most of you guys probably heard of a PCR. This was first invented, I think, back in the late 1980s. We started using it in the lab in the 1990s. But the PCR, the polymerase chain reaction, was the first reaction that we developed to quickly amplify DNA. So the concept is, if you have your target DNA here, we know that DNA is double-stranded. To amplify this DNA, we can design 2 primers, P1 and P2, and what we do is we do a step to break apart the DNA using heat, and what happens is then these primers can anneal to each end of the primer from the 5'3'. This is the annealing step, and then after that, use the polymerase to amplify it. So what you see is from one DNA, you end up with 2. And you keep on going so forth, denaturing, hybridizing, extension, so forth, and you can see this becomes the exponential amplification. So So the importance of this technology when it first came out was before it was very hard to amplify just a piece of DNA. You know, we usually have to clone it into a plasmid or take a larger piece of DNA and try to amplify in bacteria and things like that. But with the advent, with the invention of PCR, we now had a way of amplifying DNA, whatever DNA we want, the size and so forth, to able to amplify DNA to study. Now, what we did after this is we took this and we converted it into what we call a real-time PCR application. So real-time application is essentially a extension of the PCR, but the most important thing is you can use real-time PCR to actually measure gene expression levels. So you can actually quantify messenger or message RNA levels in biological samples. And what typically we do is we monitor some kind of fluorescent recorder or something that's incorporated into the technology to see when and how often a piece of DNA is amplified. So this is some forms. This is just an example of some types of PCR reaction we do. We have the cybergreen, TACMen, molecular beacon, bicycler. What typically we use is the cybergreen and TACMen data, and I'll show you some examples of how this works. So cybergreen is essentially a system where in the PCR reaction, first we have the denaturing step, and what happens is when you anneal your primers and you start extending, these green things, which is a fluorescent probe, gets incorporated into the DNA. So what happens is you can start measuring how much DNA is made by measuring how much of these fluorescence is incorporated. So instead of running a gel at the end to see or trying to quantify how much DNA is actually made, you can actually use the fluorescence label and measure the fluorescence to see how much amplicon or how much DNA you actually amplify. And this was cybergreen and is made by Now, this is the TACMen protocol, and this is done by ABI, and this is a little bit different than the cybergreen we talked about. Instead of having a probe, what this method uses is the primer that you use to generate the amplification has attached to it this reporter dye. And so what happens is when you start doing your annealing and your polymerase start extending your DNA, when it reaches this primer, it releases this reporter dye. And so what you do is then whatever is released is then measured by the instrument, and that's how you measure amplification. So why is this important? Well, if you look at PCR, this is typically what we see in a PCR result. And what you can see is when you start off, you have low level of DNA, and it's not measurable. Once you do an amplification, that's one cycle, and you keep on going forth until you get to a point where you see the fluorescent probe start to increase. And what you see is you calculate how many cycles does it take for the amplification occurs before you can actually measure some DNA amplification. And this, by using this, you can then correlate to the gene expression level. So to give you some examples, here you can see if you take multiple genes and you do an RT-PCR reaction, you can see that they all amplify at different regions. You can see that this sample here, they all have starting out the same starting material, but you can see that this sample amplifies at an earlier cycle, where this one amplifies at a later cycle. And by calculating at how many cycles does the expression start to pick up, you can go back and extrapolate to determine what is the gene expression level of each. So the advantage of this is now we have a technique that we can do in lab to measure what the gene expression level of a specific gene is. Now, is this useful? Well, there are some clinical assays based on the RT-PCR that are currently being used, and you guys probably have heard of most of these. We have the Oncotype DX, which will be used in colorectal cancer and breast cancer. We have Carcinogenesis, which is used for adenoma unknown primary, and Rosetta Genomics, who actually uses microRNA. But all these clinically applicable assays we are currently used that are now being used is all based on the RT-PCR assay. So we're going to talk a little bit about these. Has anybody ordered the Oncotype DX before? All of you guys? Okay. For breast, colon? Okay. For breast. Carcinogenesis, has anybody ordered that for adenoma unknown primary? Ben has. And I doubt anybody has ordered the Rosetta Genomics. Is that correct? Okay. So this is where I'm going to start deeply and try to make this a little bit more clinically relevant for you guys. And then we're going to talk about the Oncotype DX a little bit more in detail. Being a GI oncologist, I'm going to naturally talk about the GI Oncotype DX because I think that is a test that is little more or less used than the breast Oncotype DX, and there's a little bit more controversy and things you guys have to be a little bit more aware about. So has anybody ordered the Oncotype DX for colon? No one. You can't do it? Okay. Is there any reason why you can't do it besides you can't do it? Okay. So let's talk about the Oncotype DX. So first of all, what is the Oncotype DX? So as you guys know, the Oncotype DX is made by Genomic Health, and it is an assay that they develop to determine if they can identify patients who are at risk for early recurrence after surgical resection. So this is for primary colorectal cancer, mainly stage 2, but their assay did include some stage 1, stage 3 patients. But the question here was, can you develop a predictor that can determine which patients are at early risk for recurrence? And if so, if you identify those patients, what can you do for them? So the Oncotype DX, in their initial study, actually took 4 large studies. And this is actually very important because when you try to develop genomic assays, you want to make sure you have a large discovery data set. And so here, what they did was they took 4 large data sets, NSBP, CO1, CO2, which had 270 samples. This is actually the Cleveland Clinic. This was not from a clinical trial. This was the biobank from the Cleveland Clinic, which had 765 samples. The NSBP, CO4, CO6. And what they did was they combined all this study to have a discovery data set of essentially over 1,000 samples. So you had over 1,000 samples, and you need a clinical outcome of these patients. So typically with early stage colorectal cancer, we tell patients that if you resect it, you have anywhere from a 25 to 30% chance of recurrence. Now, that varies depending on the stage, but that's typically the number we quote people. So we have the clinical outcome of this case, of these data sets. We know about 25 to 30% of them recurred, 70% of them did not recur. So what they did was they compared the gene expression data between those who recurred and those who didn't recur and asked the question, are there differential gene expression between those 2 subgroups? And I'm making this sound simple, but they also went through a lot of analysis and algorithm and a lot of gene expression data analysis that we're not going to talk about today. But essentially with that, they came up with their final list that I'll show on the next slide. And after that, they validated on this large Quasar study, which is the clinical study of the stage 2 colorectal cancer. Okay? So what genes did they come up with? Well, this is their predictor. It's a 12-gene Oncotype colorectal cancer. And I think there's a couple things to mention here. They have 3 genes that they think is from stroma, 3 genes that is from cell cycle, and GAD45, which is not really stroma or cell cycle dependent. So these are actually their 7 genes that determines the recurrence score. What they also have in this is these 5 genes, which is called the reference gene. And this is actually a very interesting concept that Genomic Health has done in their assay development. Now, typically when we do experiments in a lab, and we say, well, what is your baseline reference gene? A lot of us will say, well, we use GAPDH or beta-actin because we use those as control and they shouldn't change. However, what Genomic Health decided to do was, well, they didn't want to use common things like GAPDH or beta-actin. They said, we actually want reference genes that are now differentially expressed in colorectal cancer. And they actually did this for the breast cancer type assay too. So they went through all their colorectal cancer specimens and came up with these 5 genes that was essentially expressed the same throughout all the samples. And so this became their 5 reference genes. And now you can see what the powerful of these reference gene is now that any colorectal cancer specimen you get should have a baseline that is stable or the same for all samples because these reference genes are all colon cancer specific. So this is probably one of the smartest things Oncotype DX did. Instead of relying on GAPDH or beta-actin like other people, they actually developed reference genes that are specific for specific cancer types. So based on these 12 genes, based on these 7 genes, they came up with this reference score. Now don't ask me how they came up with this formula. Again, this was their way of doing their gene expression analysis. But essentially what they did was they came up with a recurrence score based on this formula where they can calculate the recurrence score of each individual sample after resection. So how does this work? Well, in order to see if the database, if a predictor works or not, you got to actually validate it on a large clinical data set. So what they decided to do was validate their predictor on the QUASAR study. So the QUASAR is an adjuvant stage 2 study that was mostly stage 2, 91% stage 2, mostly a little bit stage 3. Most of these were colon cancer patients, but there was some rectal, which is important because, as you guys know, colon and rectal are different biologies. But their question is, based on this study, we know some of the patients got 5FU, some got observed, but we all know their clinical outcome, which is whether or not they recurred or not. And the question is, can their predictor predict which patients are higher risk for recurrence? Okay? So again, here's their recurrence score. Here are the genes and their recurrence score. And what they did was their recurrence group, when they calculated the score, they came up with 3 groups. The low group, which has a recurrence score less than 30, intermediate score of 30 to 40, and high of greater than 41. And when they applied this to the QUASAR study, they saw that 43% of the patients are in the low group, 30.7% of patients are in the intermediate, and 25 are in the high group. And when they looked at the Kaplan-Meier curve, what they showed was that those who were predicted to be low had a much higher recurrence-free survival than they predicted with low. So they showed, for the first time, based on an RT-PCR assay, that they can develop a predictor that is clinically applicable that was validated in a large data set. Right? Now, the things you got to be asking yourself is, well, this is fine, but how good is this really? Here, you have this line, which is already 80% survival, which is good. Here, this blue line is about 85%. So even though these are statistically significant, how much information are we really getting out of these assays based on the score? Okay? Now, once you have a prognostic marker, the next question is it predictive, right? Because we all know if you have a patient in front of us with colon cancer and we tell them, oh, guess what? You have a high risk of recurrence. Their next question is, well, what are you going to treat me with? Right? And this is the biggest difference between the colon cancer oncotype and the breast cancer oncotype, and why the breast cancer is used more often than the colon cancer. Remember, with the breast cancer oncotype contains both prognostic and predictive information. So if you apply a breast cancer oncotype, you can identify those patients who are at high risk for recurrence, but you can also predict who will benefit from chemotherapy. The biggest problem with colon cancer, which has not yet been resolved, and which is why most of us do not order it, is it's unclear whether or not they have a predictive capability or not. So if we order it and someone comes back with a score of 45, which is high risk, we're unclear what to tell this patient in terms of adjuvant chemotherapy benefit. So now you're ordering a test that you get information, but what's unclear how it's going to guide your treatment. Okay. However, it does have some predictive information in it, and this is a graph that you guys should all be aware of. So this is a graph of patients in the QUASAR study who are broken up into the low recurrence risk, intermediate, and high recurrence risk. And the bottom line is patients who got chemotherapy, and the top line is patients who got surgery. And what you see is we see that adjuvant chemo is better in these group of patients than surgery alone. However, the question is, can the Oncotype DX actually predict who will actually benefit from surgery? Now, remember, when we have a patient with stage 2, without the Oncotype DX, if I have a patient and they have stage 2, and they ask what is their risk of recurrence, typically what we tell them, based on their pathological feature, is anywhere from 10 to 25%. Okay? What is the benefit of adjuvant chemotherapy? Well, the benefit for adjuvant chemotherapy across the board is about 20%, meaning that if someone has a benefit of about 12%, has a recurrence risk of about 12%, giving them adjuvant therapy will reduce that risk of recurrence by about 20% or 2.4%, right? So the absolute benefit is only 2.4%. However, if you have somebody with a recurrence risk of 25% because they have a higher risk factors, then the benefit of adjuvant therapy is still approximately 20%, which in a 25% patient would make it about 5%. Okay, so that absolute benefit is 5%. So when we're talking to a patient about stage 2 disease, this is one of the biggest things we're talking about. we tell them, well, your benefit is about 20%, but your absolute benefit depends on where you start out. So if you have somebody who has a recurrence risk of 15%, with adjuvant therapy, you can reduce their risk down to about 12%. If you have somebody with a recurrence risk of 25%, you can reduce their risk of recurrence down to 20%. So it becomes a conversation with your patient whether or not that absolute benefit is worth adjuvant therapy. And this is where the Oncotype DX can actually come in handy. Because what we do know is if you have somebody who has a recurrence risk or recurrence score that is high or greater than 40, the recurrence risk is actually predicted to be around 20-25% or higher. Whereas if you have a low recurrence risk, the risk of recurrence is about 10 to 15%. So this is where I actually use Oncotype DX. If I have somebody with stage 2, and I look at their path, and I say, well, your path, there's no lymphovascular invasion, there's no peritoneal invasion, you have 40 lymph nodes that was isolated, you have no perforation, you have low risk, I'm estimating your risk of recurrence to be about 10-12%. If I order the Oncotype DX, most likely it's going to be low, and giving you adjuvant chemotherapy, absolute benefit is only going to be about 2%. Is that really worth to you? And most patients will say, well, 2%, I don't want any adjuvant. However, some patient will say, well, are you really sure my risk of recurrence is only 12%? How about there's something in the biology that you don't know about? Can we order the Oncotype DX? So you order the Oncotype DX on this person, and the recurrence risk score comes back at 50. Then you know their risk of recurrence is now, instead of 10 to 12, more like 25, and their adjuvant therapy benefit is 5%. So you can see how this can guide your treatment, because someone with a low absolute benefit of 2% may not want adjuvant, where somebody whose benefit is now 5-6% may want adjuvant therapy. So this is where the Oncotype DX comes in handy, but unlike the breast, where you can just order on everybody and just say, yeah, if you come back high, we're going to treat you with adjuvant therapy because we know it works. For colorectal cancer, you got to think a little bit more and ask, is that really going to change how I treat whether or not I give you adjuvant? And because of that, that's why the colon cancer is not used as commonly as the breast cancer.
So, this is stage 2 patients with a high score?
Mm-hmm.
How many were there?
I'm sorry?
How many were there? Do you remember?
How many? If you look at this, 25%. 25%.
They look at MSI status performance.
We're going to get to that. Yeah, we're going to get to that. So just to give you some kind of data, does anybody know how many breast cancer Oncotype DX is ordered or run by Genomic Health daily? Take a guess.
2,000.
Well, they will be very rich. It's about 400 or 500. Okay, a day. Okay, so that's how much breast cancer Oncotype is ordered a day at Genomic Health. Does anybody know how much colorectal cancer Oncotype was ordered in the first year since it became available?
50.
You guys are very passionate. 2,000. Okay, and that tells you. But the reason why is most of the time we don't know what to do with the data. Okay, it's not for everybody. It is useful for certain patients that I will show you, but because it's not useful for everybody, it is not commonly used as breast cancer.
So is all the gene expression validation stuff published somewhere? Is it all?
Yes, it's all published. Yeah, there's been so let me get to that. So sorry about that. I should have made this white. Sorry about this, but now there has been 3 data sets that's been published. The first one is a quasar data set, which is published in JCO, came out about 2 years ago by Gray et al., and that was a quasar study. Most recently, there has been a CLGB 9581 study that was published about a month ago in JCO that validated a second large data set. Now, the one thing I'm not going to get to is they're actually, because it's very hard to use this as prognostic, they're actually trying to validate this in stage 3 patient to determine whether or not OX is beneficial using their Oncotype DX. As you can see, they're now trying to switch gears and say, well, can we make it predictive of OX? Now, we don't have time to talk about this, and the data's still not certain yet, but there's some data suggesting that this can be used to predict who in the stage 3 setting can benefit from OX. Now, let me tell you about the Oncotype DX, and this is what Sandeep was talking about. This is probably the most important figure out of the paper, okay? So this is the Oncotype DX. The recurrence score is here, and the risk of recurrence. And you got to be aware of three types of group when you order this test. The first group is down here, which are the MSI patients, okay? As you guys know, MSI, microcellular instability, is a measure of mismatch repair, and you know that approximately 10 to 15% of stage or early stage colorectal cancer are MSI. Not a whole bunch, but MSI, there is 10 to 15%. You guys also know that MSI are good prognosis patients. These are mainly right-sided colorectal cancer tumor, and they do very well. Okay, they have good prognosis, and typically, if you have MSI, we do not give these patients adjuvant therapy because there is no benefit, and there's plenty of data to show that. What the Oncotype DX did was, when they looked at all the patients with MSI, okay, which was about 11%, it did not matter what their recurrence score was. Okay, their risk of recurrence is less than 5%. So automatically, what this tells you is, if you have somebody with MSI, you do not need to order Oncotype DX because the data is irrelevant. Okay, you can order someone with MSI, you can order Oncotype DX, and it can come back with a recurrence score of 60, and it doesn't matter what you do. The risk of recurrence is still low, and it's not going to change your clinical judgment. Now, unfortunately, Genomic Health doesn't tell us this. There's no reflex. But what Genomic Health does is, if you send it to them, they will always get MSI. But if you don't tell them that if they are MSI, don't order the Oncotype DX, they will go ahead and order it. Okay. But for most of you, for somebody with stage 2, when we're talking about adjuvant therapy, you know that most of us will order MSI to determine whether or not they need adjuvant therapy or not.
Can this actually be done on fixed tissue after the surgery?
Correct. This can be done on FFPE. So one of the advantages of RT-PCR is they can be done on FFPE. So this is all done on FFPE. For you guys, if you want to consider ordering an Oncotype DX in your stage 2 patient, and you're determining do they really want adjuvant therapy or not, the first thing to do is order MSI. Okay. If they're MSI, no need for adjuvant therapy, no need to order Oncotype DX, you're done. Okay. Now, let's say they're MSF, which is 85% of the patients, and what do? Well, we look at high-risk feature. Here, this curve up here is patients with T4. T4, as you guys know from the Mosaic trial, is a high-risk feature, right? So the Mosaic trial gave us a list of high-risk features. T4, perforation, inadequate lymph nodes, so forth, okay? And this comes up to a board question. Those who have high-risk features have a very high risk of recurrence, okay? So in this case, it doesn't matter what your Oncotype DX is. You already decided that this group of patients, you're going to give adjuvant therapy already. So in this group of patients, no need to order an Oncotype DX. You almost made the decision you're going to treat them adjuvantly, so getting an additional test is useless. Okay? So the patients that's useful is this middle group here, T3 and MSS, which is 75%. What you can see here is this group of patients here who had the high-risk recurrence score. Okay? These people who have a high recurrence score mimic those patients with high risk that would typically give adjuvant therapy too. Unfortunately, the problem is this is out of the 74%, this is about 20-25%. Okay? So the question becomes, are you going to order an Oncotype DX on every single patient with T3 MSS to capture this 25%? Okay? And some people may say yes, some people say that may not be cost effective. But this is where the decision really takes place and whether or not you guys should order the Oncotype DX. We need to capture this group of patients because these are the ones who benefit from adjuvant. Are there enough clinical data for us to suggest that we're going to treat them with adjuvant therapy, or do we need more information? So if I see a patient with stage 2 and I review all their records and I review everything with them and I said, I think you have enough high-risk feature that I'm going to give you adjuvant. I don't even order the Oncotype DX because I made the decision I'm going to order the Oncotype DX. It's not going to change my decision. If I have a patient that I'm unclear and say, well, I think we need more data, let's order the Oncotype DX to get more data, and let's make our decision. So I think that's the biggest difference between the colon cancer and the breast cancer. The breast cancer, you order to make a decision. The breast cancer comes back high, you're giving adjuvant therapy. The colon cancer is used as an assay to give you more data and more information, and we incorporate that into our decision-making decision.
How do you handle intermediate practice?
Which
How do
one?
you handle intermediate risk?
Yeah, so that's,
What are your tiebreakers?
yeah, so again, the problem, and for those of you who've been in the clinic with me, stage 2 patient is a 45-minute conversation on whether or not you need adjuvant therapy. So unfortunately, it's hard to say a general guideline because every patient is different. So when you see a patient with stage 2, you have to take into consideration what is available to you, what the patient wants. That's a very critical thing, right? We all say patients with breast cancer, if you tell them there's a 3% benefit, everybody wants adjuvant therapy. Colorectal cancer is not, it's not as convincing. You can tell a colorectal cancer patient, you have a 5% risk of reduction with adjuvant, and they're going to say, I don't want it. And I think one of the reasons is our stage 2 colorectal cancer patients, our average age is much older. So if you have a 70-year-old and you tell them, yeah, your risk of recurrence is 25%, I can give you adjuvant, it's going to reduce it down to 20, they're going to say, well, I'm 70, I have diabetes, I have heart problems, do I really want 6 months of therapy? So what is the risk of death from 5-FU therapy? Anybody know? It's about 2%. So in the middle age, is that even worth it? So the problem with the intermediate risk is, unfortunately, there's not a one defined algorithm. It is a long conversation looking at everything and deciding what is the true benefit for a patient. Okay? So I'm going to briefly talk about the oncotype 20 for the breast because this is the RT-PCR, but you guys know about this. This is a 21 gene, similar to the colon. They have low risk, intermediate, high risk, and they show in their data that it is predictive and prognostic, which is why it's being used in clinical, and a lot of you guys order it. Now, one other important thing that I showed you with the colorectal cancer, which is very important, is everything I've shown you so far has been a retrospective analysis, right? It's all been retrospective validation. They've done 3, and they've done a great job. They did 3 large data sets, and they've all been 1,000 plus, but the problem is it's all retrospective. There is a difference between retrospective and prospective, and the difference between the colon and the breast is the breast has a prospective trial. So there is a Taylor X trial which uses the oncotype DF to randomize patients based on the recurrence risk to determine what kind of adjuvant therapy they get. So in order for a biomarker really to be clinically applicable, it has to be done in a prospective manner, and this just shows you how far ahead the breast is with the colon. To finish up the RT-PCR, I did mention there are other RT-PCR assays out there. This is a biotheranostic, which is used to do cancer of unknown etiology. This is based on FFP and RT-PCR. The Rosetta Genomics is microRNA. Password is actually a microRNA, but I just threw it in there because it's adeno or unknown. The thing I want to mention about this is you look at the sensitivity, okay? They claim this overall sensitivity to be 86%, 90%, which is very high. What you guys got to be aware of when you order these tests is if you have adeno unknown primary and you say, someone tells you, oh, you can use the biotheranostic because they got sensitivity and specificity of 90%. What you got to remember is all these studies were done on well-differentiated adenocarcinoma, okay? And our pathologist will tell you, well, if I have a well-differentiated adenocarcinoma and I look at it with my eyes, see, I have a sensitivity and specificity of 90%. When do we have problem with adeno unknown? When it's poorly differentiated, okay? If you look at their data and look at poorly differentiated, their sensitivity and specificity go down to 60%, okay? But what they tell everyone is their sensitivity and specificity overall is well differentiated. So a lot of community oncologists, we see a lot of this adeno of unknown, they automatically order the biotheranostic to do adeno of unknown because they think it's going to help them a lot. But sometimes when you interpret this data, you got to be a little careful, okay? So I'm going to switch gears now and talk about microarray. Okay, so board question number 2. 44-year-old now undergoes emergent appendectomy. Pap reveals appendiceal carcinoid tumor, size 1.9 centimeter. This is for a second-year fellow. Who do we have? What do you want to do for this patient? Which second year do we have? Second year here? He's on the second year. Yeah. What do you want to do, Sunny? What do you do? And this was on my board. This was the actual question on my board. Yeah. So Sandeep is on the right track. The third year wants to handle this. What does the third year want to do? Yep. Yep. Yeah. So the answer is observation, right? So what Sandeep is trying to bring up that he can't remember is for appendiceal carcinoid, the size cutoff is 2.0 centimeter. And the reason why is there was large studies, actually not large, there are only about 40, 50 patients, but there's studies from the Mayo Clinic that looked at outcomes of resected appendiceal carcinoid tumor. And what they found out was if your tumor is less than 2 centimeters and you do a just observation from appendectomy, your risk of recurrence was less than 2%. If your size is greater than 2, then greater than 3, your risk of recurrence goes up to like 20%, 30%. Okay, much higher risk of recurrence. So anytime you see appendiceal carcinoid and the size is 2, and the magic number to remember, unfortunately, is 2, because that's what they decide to do when they look at their study. If it's greater than 2, you need to tell your surgeon to do a hemicolectomy. If it's under 2, you can just observe and watch them, see what happens. So the key point is, again, for the board, know your indications for surgery, because a lot of the management you see on the board will ask you questions like this on what you should do next. Okay, so let's talk about gene expression profiling, which is a lot of what I do. So gene expression profiling is methods capable of giving a snapshot of RNA expression. It can be used as diagnostic. And the important thing is it shows how RNA levels can change and provides large amount of data. Okay? So I'm not going to tell you guys how to analyze microarray analysis, but I just want to give you a brief overview of what it entails so you guys know what microarray data is, because it's not as prominent as it was 5 years ago, but we still see a large amount of microarray data. So there are two types of microarray data. There's what I like to call the two-color, which is the adjuvant, and there's the one-color, which is the asymmetric. Okay, both are well used, and both are used throughout by everybody. We tend to use the asymmetric here at Duke. Down at UNC, they use the adjuvant. Well, what's the difference? So this is the adjuvant, and in adjuvant, the way this microarray works is you actually have two phenotypes. Okay, so you have, let's say, for example, untreated cell, and you have treated cell, and you isolate the RNA from them. Okay, what you do is for each individual one, the RNA is labeled with a different color. And so when you hybridize the two different colors, what you're trying to figure out is one color more overexpressed or more abundant than the other. So you can imagine if something in your treated cell is more expressed and has more of this fluorescent color, then the red one's going to have a higher intensity than the green one. And so in the adjuvant, the two-color one, you label your two phenotypes. Okay? How is this different from the asymmetric? Well, with the asymmetric, you don't have two phenotypes. You have one phenotype. And what you do is you just take cells from whatever things you want to study, and you do all these steps. But what happens is you biotin label the transcript of interest. Okay? And then what happens is this biotin then gets hybridized to an array. Okay? And then based on how much of this is hybridized to array, will give you a signal based on how much of it hybridized to a specific program array. So this tells you based on how much hybridization you get on the level of how much RNA you have at color intensity. Okay? We don't need to talk about this. But essentially what you get is, so in the two probes, remember there's two colors. That's why you get these pretty green, red, yellow. Okay? When they're neutral, they turn green. If they're up in one, red is up in the other, yellow. If there's not a lot of that probe, it's black. Whereas in the one probe, what you see is one color and different intensity, whereas here, it's more intense than this spot, which is black. Okay? So the question is, how do we go from here to actual gene expression data? So then this just shows you more detail about the red, yellow, and green. But essentially what you end up with is this. Okay? So you get a bunch of pictures, and what you do is we put it in the scanner, and the scanner actually measures the intensity of these probes. And then through methods that we don't need to discuss here, what happens is you get gene expression level, because as you know, there's no way we can analyze fluorescence or colors, right? What we need is actually quantitative numbers, and so all those fluorescence or color gets translated into actual quantitative numbers. And so here, these are actually the asymmetric control probes, and they use these as control. But what they do is everyone has a gene expression level. And what we do is then we use these gene expression level to analyze to determine if there are differential expression between two phenotypes. So the biggest thing about gene expression is if you want to see if a gene expression is high or low, you can't do it in one sample, right? Because you can't really compare what's high or low between one sample. You actually need a group of samples, and typically what you have, when what you hear us say, is you have two phenotypes with two classes, right? For colorectal cancer, that's that we have patients who recur versus who don't recur. Those are your two phenotypes. Once you have two classes, then you can actually do the gene expression and using different methods, compare the differences between those two classes to see if a gene has differential gene expression. So the most important thing to remember about gene expression is you need to have at least two phenotypes. Whatever two phenotypes you want to look at, whether it's drug sensitivity, recurrence, survival, it doesn't matter. But to compare the expression of a gene, you need to have at least two phenotypes. Okay? So does this work? Well, this is one of the first paper done by Todd Golub, where he actually used hematological malignancy. This was AL, AML. This was published in Science back in 1999. And you can show that by gene expression profiling, you can use gene expression to show the difference between AL and AML. Okay? You guys seen plenty of these heat maps. This was Chuck Peru's, where he used it for breast cancer. You know, I could have usually used Sandeep Dave, as most of you guys know him here, and shown his lymphoma work. Okay? So gene expression is honestly getting, as far as Matt will tell you, is getting out of style. A lot of people don't use it as much. We're all moving off to sequencing. But we are still using gene expression to classify tumors, and the question is that clinically relevant? Well, I'll show you two examples that's kind of being used, and it's called a mammoprint and coloprint, and this is probably the equivalent to the Oncotype DX in breast and colorectal cancer. Now, this is mainly used in European, and I think this is very interesting, because in Europe, with gene expression, what you typically need, unlike the RT-PCR, is fresh frozen sample. With the Oncotype DX, RT-PCR, you can use paraffin, and you can get paraffin anywhere here. With fresh frozen, it's a little hard to get, but in Europe, apparently, it's a lot easier to get, and as a result of that, they were able to develop these gene expressions for breast and colorectal cancer. So mammoprint can be a breast cancer, early stage, and what they did was based on whether or not they recurred or not, just like the RT-PCR, they developed a gene classifier. They developed 7 genes, which is prognostic, and if your sample had this phenotype or represented this type of gene expression, you were a good signature, and if this is like this poor signature. So very similar to the RT-PCR, except in the Oncotype DX, they developed this quantitative score to develop a recurrence score to say whether or not you're good or bad. Here, if you have a sample, what you're doing is you're comparing the gene expression level or the gene expression matrix to see does it resemble this or does it resemble this. If it resembles this, then it's in a good prognosis. If it resembles this, it's in a bad prognosis. So this is their data, and so you can see if it's a good prognosis, you can see these patients do well. If it's bad prognosis, these patients do very poor. So just like the RT-PCR, same concept. And this is their low risk, this is high risk, and their benefit for adjuvant therapy shows that patient with high risk benefits from adjuvant, those with low risk did not. So just like the RT-PCR, you can use the gene expression because it's the same concept to show that it's prognostic and predictive. And again, they're done in Europe, just like the Taylor X, they have a study which actually looks at a prospective analysis of whether or not you can use this to make a clinical decision. So the difference, gene expression, you get a lot more data because you're looking at a lot more genes. RT-PCR is limited data, but again, they're very similar concepts because they're using gene expression. Europe likes to use the gene expression because they can get access to fresh frozen tumor samples to get the RNA. We tend to use the FFPE, which is more the oncotype. So in Europe, the mammoprint and the coloprint. Here we use the Oncotype DX. So the coloprint is essentially the same thing. This is the Oncotype DX equivalent, and again, they show that if you have a good prognosis, you do well. If you have poor prognosis, you do poor. So this is just the same as Oncotype DX, except this is done in Europe. And they actually have a prospective trial, which is very interesting. This trial is actually going on here in the U.S. But similar to the Oncotype DX, the coloprint is only prognostic and not predictive, so you can't do a prospective trial to determine whether or not you can make treatment decision. So what they decide to do was they actually have a trial here, which is a prospective trial, but the outcome is how physicians use this data to decide whether or not they're going to change management, right? So we kind of talk about with Oncotype DX, we go through all this thing to determine whether or not we should use this and if they're going to change our treatment. Well, they have designed a trial to see if a physician will actually use this test to change treatment decisions. So again, this shows you that the colon cancer equivalent is not predictive, it's only prognostic, but this is a prospective trial that will determine whether or not physicians will actually use this data or use this assay to change treatment decisions. Okay? So let's finish up with sequencing, which is by the hot thing going on right now. And we'll end up with our last board question, number 3. So, this is for a third-year fellow. 34-year-old female with early-stage colorectal cancer, found to have MSI. Further test reveals a BRAF mutation. Further workup for Lynch syndrome should include 1, 2, 3, or 4. We have a third year. Nothing. She does not have Lynch syndrome. Very good. Why?
What I want to say is that that's what the algorithm says.
That's what the algorithm says, okay. Perfect. It's because of the BRAF, right? So what is Lynch syndrome? Lynch syndrome is characterized by deficiency in mismatch repair, right? So, as I said earlier, about 10 to 15% of early-stage colorectal cancer have a deficiency in mismatch repair. Approximately 10 to 15% of patients with MSI have the Lynch syndrome. The question is, why do you have the deficiency in mismatch repair? Is it acquired or is it congenital? Okay. So what can cause mismatch repair deficiency? Well, BRAF mutation can. So if you have a BRAF mutation, your mismatch deficiency comes from the BRAF mutation. Therefore, it is a acquired mutation and not genetic.
So are there no patients with families who they don't end up with BRAF mutation?
Yeah, no Lynch patient. No.
So why is that BRAF located?
That we don't know. That we don't. Why BRAF causes a deficiency in mismatch repair, the mechanistic of that is what remains unknown. Correct, but you also have to be careful about MSI and stage 4 disease, right? So 10 to 15% of patients have MSI, which is good prognosis. If you look at what percent of stage 4 patients have MSI, it's less than 2%. So the majority of patients who have MSI will not metastasize, which is why MSI is such a good prognosis. But this answers your question is, this kind of, is leading to what you're asking. Why is that subgroup of MSI who metastasize, which is very small percent, 1 to 2%, has such a worse prognosis? One, because they're already metastasized, and is their underlying biology different that causes them to have a worse prognosis? And that we don't know.
Well, it's also interesting that you would never see that in patients MSI patients, because there's a syndrome, there's those patients that get 1,000 mutations.
Correct.
Across, presumably some of those are drivers.
Correct.
It could be just if you end up with that, and a tumor with them already with Lynch syndrome or MSI,
is it breast damaging? Correct. Yeah, it could be. Right. Correct. But then that wouldn't be a genetic mutation. That would be an acquired mutation, right? And so then, but you're right. So the question then becomes, you have a genetic form of colorectal cancer, but did you acquire a driver mutation that makes it a worse outcome of so is that really a genetic hereditary, tumor because you acquired a driver mutation, right? So the important thing about this, and this is going to be all over your board, is know your genetic mutation. Translocations, mutations, there's going to be 10-15% that's going to be all over your board, so know these for your board. So, let's finish up with sequencing. How many base pairs on the human genome? Matt?
Genome run.
Huh? Matt, you should know this. 3 billion. Very good, 3 billion, see? How much does that cost to sequence per human genome? 2.7 billion. How long did it take?
12 years?
13 years. What was the first human genome sequence? When was it first completed? 2003 was when the Science Nature paper came out. So, because of this is why sequencing has not been used recently until now, because now the cost of sequencing is so much cheaper, right? 2.7 billion to do first human genome. The cost to do a complete genomic sequencing is anywhere from 10 to 15,000, and you can do it in weeks, okay? Which is why sequencing is moving to the forefront of how we characterize tumor biology. It is no longer cost dependent or time dependent because we can do it much cheaper and much faster, okay? Now there's a couple of next generation sequencing. There's a couple ways to do it. We'll briefly go over this, but these are the sequencing platform, and Matt can probably tell you more about these than I can. But there are Illumina, ABI Solid, Roche 454, and the new one, Polator Helixscope. Okay, the ones that are most commonly used are the Illumina, Solexa, ABI Solid, and the 454. These are the more commonly used sequencing reaction. So how do these work? Well, one is the bridge PCR, which is used by Illumina and Solexa, and what happens is you have these linkers, and these DNA fragments are attached to these linkers, and they kind of bridge over, so you get data from both ends. And what happens is these get amplified, and after they get amplified, they get released again. You can sequence this. Okay, so this is what we call a bridge PCR reaction to sequence your samples. Okay, emulsion, which is done by 454 and Solid. What happens is these linkers or these pieces that you want to sequence get emulsified along with these linkers, adapters, and then they attach, and these are what gets sequenced. So very similar technology. Essentially, you want to get fragments of DNA so you can sequence them. Okay. I think what's important for you guys is how to interpret the sequencing data. Okay. So this is Sanger sequencing data. This is what I did when I was in grad school. And when you do Sanger sequencing data, there's no doubt what you get when you sequence, okay? So you sequence it, and you get A, C, G, T, and when you're reading it, you read it C, 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 G, 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 T, A, C, C, G, A, and there's no doubt what you get is what you see, okay? And that's why Sanger sequencing, whatever you see is what you get. With next-gen sequencing, what happens is what you get is the hybridization or the fluorescence is determines, and the peak determines what kind of sequence you get, okay? So there's two concepts here. One, if this has a high peak and, it's clearly a C, then you know what your sequence is. The problem is when you get these lower peaks or mixed, the question is, what is it? Is it really a C or is it a T? So unlike the Sanger sequencing, where this is clear-cut as, ACGT, next-gen sequencing or any of these newer sequencing, you got to be a little careful how you interpret the data. The second part is when you look at next-gen sequencing, what's important and what you hear us talk about is coverage and how much of it do you read. So what you can see is here in Sanger sequencing, you're reading one thing. You're reading one sample because you know what it is. Here, what ends up happening is you get multiple reads that span a certain area that you want to see, and depending on what your read is, you got to determine whether or not that is a mutation or not. Okay? So this is my way of trying to show you guys how sequencing data works. So let's say you have a consensus sequencing, right? We all know you have sequence 5', 3', U', and you read it. Okay, so this is the sequencing you're reading for. You do next-gen sequencing, what happens is you get a whole bunch of these reads, tiny reads, because remember, these are fragments of DNA, and what we do is we align it to our sequence. So I make this a small case, but here, for this T here, we have 6x coverage because we have 3 sequences or 6 sequences that align to this read. So you have 100% identity, okay? So this is what people mean when they say the amount of coverage you need to make sure your sequencing data covers everything. Now let's look at a mutation. Well, let's say we want to look for a mutation and we see the C is changed to A, and we do our coverage and we get 6 reads that align to it. So you have 6x coverage, but out of the 6, only 5 have a mutation. Well, 5 out of 6 is pretty good, so we might call this a real mutation. In contrast, let's say you have this, right? Let's say this G becomes a T, you have 100% mutation, but you have only 2 reads. Oh, okay. I'll be done in like 2 minutes. You have 2 reads. So one can almost argue, well, you identified mutation. It's 2 out of 2, but you have only 2x coverage. So this is the biggest issue with sequencing, okay? All depends on how much read you can cover, because you can imagine if you have a large number of reads and more mutation, you're more likely to call this a mutation versus this where you have less coverage. So you can imagine if you do 4 more and they turn out to be G, then your mutation is really not 2 out of 2, it's 2 out of 6. So this is the biggest thing you got to worry about sequencing, and for sequencing genome in cancer, you need at least 100x coverage. So when you get all the sequencing data, what you guys have to be aware of, this is how next-gen sequencing is done, and what is the coverage, and how real is the mutation, okay? So kind of where we are in terms of sequencing, identification of new prognostic and predictive biomarker, what's going on is identification of accidental mutation, and we use it to molecularly classify tumor. So conclusions, genomic profiling is instrumental, assets are being incorporated in clinical use. Studies will be to decide how we use this data. Board reminders, know your staging system as it pertains to treatment, know your indication requirement for surgery, and know your genetic mutation. Okay? Thanks, guys.